Okay, so just one more comment about the pupil size. I want you to note, remember I did say that the iris is a smooth muscle, which means it's going to use the autonomic nervous system. Obviously, there are two parts of the autonomic nervous system. We have parasympathetic, which is rest and digest, and sympathetic. So it makes sense, think about parasympathetic. When you're resting, about to take a nap, you do not want a lot of light coming in. Okay, therefore you're going to have constriction of the pupil. Sympathetic control, fight or flight, you might be in fear of your life. You need to be taking in a lot of information around you, so you would need adequate light so your cones can really see with great clarity. Okay, just so y'all know, I'm only going to talk for about another 45 minutes. The first video was 42, and I just don't feel like it's fair to go that much further. So however I get within that time, that's about when I'll stop. So the neural layer of the eyeball is where the photoreceptors can be found. Okay, and we call this neural layer the retina. So it's only deep to the choroid. It is not found in the anterior portion of the eye, like next to the iris or the cornea or any of those structures. So the first layer is pigmented. As I mentioned earlier, the melanin in there absorbs light, so it doesn't allow it to scatter everywhere. The deeper layer is where you actually have the receptors for light waves, which are photons. And of course, all of those axons are going to end up exiting the eye through the optic nerve. Okay, so the photoreceptor cells are what detect the stimulus, in this case, light waves or photons, and they transduce or convert them into electrical signals. The ora serrata is the boundary between the anterior edge of the retina and the posterior ciliary body, and that basically creates that visible line where you will start to have the retina with those photoreceptor cells. Mentioned this previously earlier today. This is the very center of the macula. This is where we have almost all cones. So this is the portion of the eye that provides the most clarity and detail and color about what we're seeing. Okay, macula lutea surrounds the fovea. We talked about that as well. So in your eye, where the photoreceptors, if this is a rod or cone, it's going to have to connect to nerves. So where all of these nerves leave, all of these axons are going to end up forming the optic disc is what we call it. And that's basically the blind spot because you do not have photoreceptors there. So in this picture, this would be what an optometrist would be able to see using an ophthalmoscope. So you can see the macula lutea, fovea centralis. Again, that's the highest area for clarity of vision. And then you can also see the optic disc. Again, it looks different there because it's absent of photoreceptors. So there's a test you can do. Basically, you can read and follow this test. Of course, focus on one, it'll go away. You can do the same thing from a pencil, and obviously, we don't see the hole in the pencil. Okay, so our brain naturally fills in the blind spot with stuff around it, which is, of course, why you're driving, why you need to look additional ways, because it might be filling in that blind spot and showing you a car's not present when it really could be there. Okay, so these are just some kind of things I want y'all to think about, and we can actually, I would like to discuss these tomorrow. Okay, so we talked about, I talked about a lot of these structures already. So basically just review lens and some of these structures that I've already talked about. Okay, so abnormalities with the eye, cataracts. So your cornea and lens should be completely transparent for light to pass. Unfortunately, sometimes part of it becomes more cloudy. Technically, it can happen on the lens or it can happen on the cornea, and that's called a cataract. Typically, it will occur with aging. Trauma to the eye or head can cause this, exposure to UV radiation. So just like you need sunscreen on your skin, okay, you need appropriate sunglasses that actually block the harmful rays as well. Diabetes affects vascular vasculature everywhere in the body. So if the rods and cones aren't able to get what they need, of course, they start to degrade, and that can lead to blindness as well. Unfortunately, they can't do anything with cataracts. They can replace the entire lens. Sometimes they'll take LASIK and just try to remove some of the cataract portion. Okay, I've already talked about chambers, so just make sure you review that and make sure you understand what fluid is on where. I talked about a lot of this already, so again, I just don't want to waste time with stuff we've already discussed. Of course, you can, read this further, again, review, come with any questions tomorrow. Review questions are always great. Okay, so now we're not so much into the anatomy, we're more into how does it work. Okay, so of course, vision is how light is reflected, how it bounces off of different objects. And so all the information that's taken from the optic nerve, of course, eventually goes to our central nervous system, the brain, and that's where we determine all the information about whatever we're looking at, size, shape, color, etc. We can also figure out how far it is, maybe how fast it's moving, the direction. And there's a lot of other senses that we use to add this information into what we're seeing. Okay, so energy exists in a lot of different wavelengths. It's in electromagnetic radiation, which are measured in nanometers. Basically, the only types that we can see are what we call visible light. So that's your rainbow. You ever heard the Roy G. Biv? For the colors that we can see: red, orange, yellow, green, blue, indigo, violet. And of course, with visible lights, depending on the color depends on how long the wavelengths are, and we'll look at a graph in a minute. So again, the unit of light, how light travels, is what we call a photon. So we cannot see really, really short wavelengths. We cannot see gamma rays or X-rays or radio waves. We can, however, see the longer wavelengths in visible light. Okay, so this is our spectrum. Okay, so again, you see basically just this narrow area is all that we can see. Again, there are the colors. So your longer wavelengths are going to be very bright colors. The deeper colors, again, are going to be shorter. Don't worry about actually memorizing any of these nanometer lengths or anything like that. Okay, so for our retina to be able to transduce or convert these photons to electrical signals, again, the light has to be refracted, or bent to hit directly on the back of the retina. So, of course, like I said, air it's not dense, so it doesn't really bend light. Water is denser, so it can refract light. And of course, you can see this demonstrated, okay, by this person in the pool here, by the degree of the angle. Of course, it looks like, he doesn't have a head over his body, but it's just the fact that the light waves are being bent, so it distorts, okay, that image. What we also have to consider about bending or refracting light is what type of surface is the light going to be hitting. So basically, convex versus concave. It's probably easier if I just go ahead and go to the picture. So this is what we call convex. Okay, it's the curved surface. So notice that the curved surface will make it converge to a focal point. Okay, when it's a concave, so I remember a cave is something that goes in, so that's why that curvature is that way. It's going to spread out the light, so they do not focus. So of course, the type of the shape of our lens, of course, the outside as light hits it, it's going to be this convex surface. So clear vision, again, light rays have to be focused on the retina. So by focusing, they have to come to one point and they have to be absorbed. Okay, you can't have light bouncing around everywhere. Again, most of the bending power, about 2/3, is from the cornea. And remember, that is fixed. Okay, and it actually doesn't bend it a whole bunch. It's closer to water. The lens, again, has the ability to flatten more or to bulge, so it can change the bending of light. Emmetropic state basically means the eye is relaxed, so we're looking at things from a distance. Okay, so normally when you're looking at a distance, your lens is going to have this more flattened shape. Okay, so it's not bending a lot. The closer something becomes to our eye, our ciliary muscles have to basically bend the lens. So it's going to have this more bulging shape to it, so it bends and converges more. Okay, if it's like this, it's going to take longer to converge. Okay, so what we can see here is they're basically showing how the light waves change as they're bouncing off an object and how that's different if you're close up versus far away. And then again, this is showing you the more flattened look of the lens when looking at something from a distance versus if you move something close, how it's going to be more bulging or rounded. Okay, so again, what controls this? I briefly mentioned this in class earlier, but this is really important. It's the ciliary body, okay, which is surrounding the lens. Remember, the ciliary body, along with the iris, are the two smooth muscles of the eye. So again, our eyes get more tired when we're looking at things close up, which means that this body is relaxed, or the muscles relax when we're looking at things that are far away. So basically, as the ciliary body relaxes, those little suspensory ligaments that connect the lens to the ciliary body are basically being stretched, so they flatten out the lens. Okay, so here it's just showing when this is relaxed, these ligaments stretch out, and so the lens is going to be, again, more flattened. This is that more rounded view. So when the ciliary body contracts, looking at stuff closer, it moves closer, the ligaments are basically loose, and so the lens will naturally start to bulge and get more of that rounder shape, which is what we see here. And of course, as I mentioned previously, these are relaxed, okay, so it's bulging, but as we age, the ability of the lens to bulge decreases. Okay, something else that's really important, again, is that to really be able to view things appropriately, to figure out movement, how close something is, you need to be able to use both eyes. So we have convergence, which happens with this too. So convergence allows the both eyes basically to meet and focus on an object. Okay, and the idea again is that we want most of these rays hitting the fovea, which is the area where we have the most cones in the eyeball on the retina. Okay, well, sometimes our eyes are not able to refract light properly to have the light focus perfectly on the back of the fovea like that. Again, this can happen just because the lens loses shape as we age, or maybe someone's born with the eyeball that's a little too short or too long. Okay, so that's going to make the focus point be in front of the retina or behind. So just a few terms here. Near point accommodation is the closest point at which the eye can focus on an object. It's about 2 inches away. Of course, as you age, things get blurrier, so we have to continue to move further away. Presbyopia is when the condition, basically you have to have something 10 to 20 inches further away. And again, this happens aging 4th or 5th decade. They have difficulty reading. They have to pull books and stuff away or put on reading glasses, things that are basically going to help bend the light properly, okay, bend it more, so it will come to the focal point in the correct place. So we call that presbyopia. Other problems we can have. So, and I know I can't draw this perfectly, but the lens should, and that doesn't look perfect, the lens should have a nice, perfect curvature to it. Okay, sometimes we can get these area, and actually it could be in the lens or the cornea, that's more flat. And that's when it causes an astigmatism. And the cornea, it can easily be fixed with LASIK, and the lens, it's a lot harder to correct. there. There's a test you can look up online if you'd like to see if you have astigmatism. It's just going to have like these little lines that kind of extend out from the middle. I know I'm just doing a rough drawing. And like most eye tests, you want to cover up one eye and then the other. All the lines should look equal thickness. Okay, if some look a little thicker than others, then you have an astigmatism. Slight astigmatism is relatively normal and not a big deal. Emetropia is when the length of the eyeball is basically perfect. That's what you want. Okay, that's when it's going to bend and refract the light appropriately onto the retina. So that would be someone that has normal vision. Okay, so again, what we want is an emetric eye. Okay, one that perfectly focuses on the retina. So both the ability of the cornea and the lens are able to refract light appropriately. We're about to look at some examples that are irregular, and we can see how we could fix that situation. So hyperopia or farsightedness, the eyeball is too short or the cornea can be too flat. So basically the lens cannot bend enough, okay, become thick enough, so the light does not focus where it should. Okay, if this is our retina, we're going to have all the focal points coming behind it. Okay, if this is our eyeball. So what we do to correct that is we put a convex lens in that bends the light more. Okay, so now it's going to come to the focal point perfectly on the retina instead of behind like it did before. So these lenses can be added with contacts, glasses, or they can even put artificial lenses insert in your eye, or LASIK to reshape. Okay, so again, this is kind of the picture you see uncorrected. It's focusing at the wrong point, so the person's going to have very blurred vision. With the corrective convex lens, it focuses at the perfect point. Okay, so it's basically your optometrist's job to figure out what strength of lens you need, okay, to get that perfect focus. The opposite of that is myopia or nearsightedness, condition where distance between the cornea and lens is too great. This is typically much more common, especially in younger individuals. So in this case, the lens is unable to flatten enough, and so concave lenses are going to correct this. So basically, myopia nearsightedness means you can see things easily close up. As things get further away, they become blurry. Versus presbyopia, which we just talked about, you can see things clearly up close, but they're going to be blurry at a distance. Okay, so again here, it's too long, so it doesn't come to the correct focal point. So we're going to add a lens that's going to bend these light waves more, again, so it comes to the perfect focal point. This is actually a little bit easier to correct with LASIK and is more exact than presbyopia would be. Okay, I also talked about this. So photoreceptors, we basically have two different types. We have rods and cones. Okay, so again, cones, I want you to think C for color, and highest density resolution, think clarity. Okay, so C for clarity, C for color. And again, they're going to be highest on the back part of the eye where the fovea is located. Rods are used for low light. So basically we can see shades of gray. Okay, they do not detect color. And again, they're going to be further away from the very center of the eye, more in the peripheral portions of the eyeball. So again, special senses, we see bipolar cells, okay, are going to synapse with these specified receptor cells, which we call photoreceptors because they're measuring photons of light. And then the bipolar cells are then going to synapse with what we call retinal ganglion cells. These are basically, these axons are going to leave, and that's what forms, again, the optic nerve. Surrounding the retinal ganglion cells and some of the bipolar, you can have horizontal and amacrine cells that basically help with the image processing, help with the transduction. Okay, so what we see here, this is the pigmented layer, okay, which you can see here, that's what's absorbing light. You have your different rods and cones. They're going to synapse with the bipolar they synapse with the bipolar cells. Okay, and then those are going to synapse with the retinal ganglion. Again, these are what's going to exit the back of the eyeball. And then you have those surrounding cells that can aid the other cells in what they need to do. So the retina, even though it's very thin, it's highly sophisticated and very complex. Okay, so rods, their outer segments, basically what makes rods and cones different is the type of pigment that they contain. Okay, so rods are named so because they contain rhodopsin that absorbs light. Again, they can't distinguish between different wavelengths, which is how we distinguish colors. Okay, so that's why you can't really see color with light. Rhodopsin has opsin in it, and it has some retinol. Opsin is an enzyme that is inactive in the dark when it's attached to the retinol. So that's, again, why we are not able to see color and things like that in the dark. Retinol is derived from vitamin A, and I really didn't want to get too much into this part, into how all this changes. But basically, the inactive bent configuration is what we call cis-retinol. When enough pigment can be made, then you can activate them, but we're really not going to get into that detail. Okay, so really there's not too much difference. They do structurally look a little different. Okay, it's really just the pigments that are inside. Again, as far as the shape and how it straightens and all that, don't worry about that. Just know the major differences between rod and cones. Okay, so cones contain iodopsin, which also has retinol in it, and the protein photoopsin. Photoopsin is similar to the opsin, but it has a different structure that allows it to absorb different wavelengths. Again, the different wavelengths are color. Okay, so that's how we can distinguish that. We have 3 different types of this pigment, the photopsin, that allow us to perceive the major colors. So basically everyone has blue cones, green cones, and red cones, as long as you're not colorblind. Okay, so of course we have other colors that we see intermediate. Okay, so when you're looking at perhaps a purple color, okay, it's going to be that you're activating some red or some blue cones, excuse me, as well as some yellow. So it's the amount, how many different wavelengths of light that you combine. Okay, so again, in the absence of stimulation, for light, stimulation is, for vision, it's light. It's photons of light. So in the dark, photoreceptor cells, it's a little different, they're depolarized. Typically, we think depolarized active, but this is different. So depolarized makes us release a neurotransmitter that is inhibitory. So it's going to stop electrical signals from being sent. Okay, when we do have some light, then the photoreceptors are able to become more negative inside. They hyperpolarize. So they stop releasing the neurotransmitter. The neighboring retinal cells now are no longer inhibited, and they can start sending those electrical signals to the brain. Okay, so this is an example of bleaching. So basically and I didn't really go through all the steps, but when you stress the eye too much, you have to basically wait for it to make the different pigments again. And so this is one of those examples. You can follow this. Look at the dot at the nose for 15 seconds. Now look here, and I'll let y'all figure out what you see. And that's basically what you're seeing is the image still from those bleached cones. Okay, so as we've already talked about, the pupil does help definitely regulate the amount of light coming in. So of course, if it's really bright, we're going to constrict the pupil. Okay, so we're not going to damage cones and bleach the cones. If it's darker, we want more light to come in to try to be able to activate as much of the pigments as we can. Okay, so in the dark, if you're in a well-lit room and it's suddenly completely dark, you cannot see anything. Okay, it takes a little bit of time and you can start to see stuff the longer you're in the room, okay, about 40 minutes total, until you can really kind of see stuff around you. Again, you're really not going to be able to see information about color or things like that. And that's because, again, the rods, the pigment they make, the rhodopsin, just takes longer. And the longer it takes to make the pigment, the less that the photoreceptor can work. So with light, if you're in the dark and let's say you just watched a dark movie, you go outside, it's 2 o'clock on the afternoon, super sunny day. When you walk out, it totally blinds you. You can't see. You bleached the rhodopsin. However, cones regenerate faster. So you close your eyes or you kind of squint, you let them adjust, you let the pupil get smaller, the pigment resynthesizes, and you can start to see again. So the sensitivity is less severe with light than it is dark, and it's because of how quick the pigments can be remade. Okay, so here we're basically comparing and contrasting what happens in the dark versus what happens in the light. So remember, what happens in the dark, we're going to be talking about rods. Okay, so the photoreceptors are rod. It's going to be releasing a amino acid glutamate onto our bipolar cell. Okay, glutamate is inhibitory. So we are not releasing as much neurotransmitter onto the ganglion cell. Therefore, we're really not going to have electrical signals that are being sent. When we have light, the types of photoreceptors that are working are now going to be our cones. So instead of depolarizing, now we're going to hyperpolarize, which stops releasing glutamate, which now allows the bipolar cell, it's no longer inhibited, so it's going to release more neurotransmitter. And so action potentials can be sent, and therefore electrical impulses can be sent to the brain. Okay, talking about color blindness. I know this is kind of one of those things a lot of people are really interested in. The person basically lacks a functional gene for one or more of the cone pigments. So again, remember, you can have, there's red cones, green cones, and blue cones. So a person could be missing one cone color, or maybe they're not missing a specific color, but maybe they have half of what they should. Okay, which creates a lot of variation between the types of color blindness we have. So we see that about 10% of males will have some form of color blindness. Okay, females have it a lot less often, and that's because color blindness is what we call a sex-linked trait. We will talk more about these when we do genetics, just to kind of try to show y'all how this works. So to be a female, you get an X from both your parents. So one from mom, one from dad. Okay, let's say the C means person has regular cones, so they're not going to be colorblind. And when I have a C like that, that means the person, that's the colorblind trait. Okay, so for a girl to be colorblind, both X's must contain the colorblind trait. Okay, so this female here, because she contains one normal, she herself has normal vision. She's a carrier, so she can give the colorblind trait to a son. Okay, but she herself has normal vision. Males only have one X chromosome. You do not have any information about color blindness on the Y chromosome. It's much smaller. So it's solely determined by what they get from mom. So if the boy gets the normal C, he's going to have normal vision. If he happens to get the colorblind trait from mom, then he will be colorblind. Okay, so that's why you see a lot more frequency in males than females. Already said this. Okay, so the most common types of tests that we use for these is Hara plates. Their collection basically of dots. We kind of saw that earlier with numbers or letters of different colors in there. And again, they have several different plates, so you can detect the degree of color blindness that a person has. Okay, so the last thing we need to talk about with vision is the pathway. So again, for every special sense, you'll need to know from the cranial nerve to the parts of the brain, what is the order that those electrical signals pass. Where in the brain do we interpret, okay, what we're actually seeing. So structures in the visual pathway are within the visual field. And basically that's what you can see on your own, okay, with no aided help with both eyes, okay, assuming both eyes are working properly. And so again, what we're going to be focusing in the center, okay, converging, that's our focal point. Okay, and I kind of talked about this in class. So everything crosses over. Okay, so basically everything that I'm seeing from my right eye is going to go to the left side of the brain here. Okay, again, the stuff that's in the periphery will go to the right eye, so it actually has to cross over at the optic chiasm to get to the left side. Versus what's in the middle, or the focal point here, is actually seen by the left eye, so it will go straight back, so we call it temporal pathway, and still go to the left side. Okay, so again, the photoreceptors are on the retina, so they're going to go to the retina. They're going to be carried by what we call optic nerves. This is our cranial nerve 2, down to the optic chiasm. Again, not all pathways will actually cross over there. Some will go straight down. Then they're going to go to a few different areas. Okay, so first, these areas are going to be, sorry about that. This is the superior colliculi. So again, that's going to be where optic or visual reflexes take place. So some of the information will be dropped off at that point. Okay, some of it will go a little further to the thalamus. We call the geniculate nucleus of the thalamus, and we can assess, interpret what we're seeing there, be made aware. And then finally, through optic radiations, it will go to the last part, which is our primary visual cortex, which of course is in the occipital lobe. And again, what you have in your left hemisphere is everything from the right eye. The right hemisphere is obviously receiving all the information from your left eye. Okay, so again, basically what's in the focal points here is going to be what travels straight in what we call the temporal. So that's not going to cross over at the optic chiasm. Versus what's in the peripheral views is seen from the opposite eye. So of course that will have to cross at the chiasm. So again, some will stop at the superior colliculi here. We'll still go through the thalamus, which is the gateway, which will eventually obviously take it to the visual cortex. Okay, so you can try this at home if you want. You can get like a clipboard or something kind of dark and put it right along your nose. And just on one side of your face, let's say the right side, you shine a flashlight into your eye. Okay, you're going to see that the pupil is not only going to constrict in the eye where you're pointing it, but in the other eye as well, which makes sense. You assume if one eye is in a high light or high dark situation, the other eye would do it as well. Why that is, it's the same nerve that's controlling those pupillary muscles of the iris. Okay, we briefly discussed this as well. Stereoscopic vision, depth perception, how far something is away from you, can really only be done when both eyes are being used. So 2 eyes is what we call binocular vision. So the brain can basically compare what it's receiving from the right eye, what's in the left eye, put the information together, and figure out how far an object is. Okay, so again, being able to process what you see involves more than just basically reading the wavelengths of light. Okay, so the axons that are going from part of the thalamus, the lateral geniculate nucleus, then the primary cortex from the retina is where it's mapped. It's basically where you make the picture, and it's actually going to be inverted, okay, or distorted. We also see, or not all parts are equal. So basically the fovea is going to have more brain area, okay, figuring out in the peripheral. Why is that? That's because the fovea is where all our cones are, which actually give us the most detailed information about what we're seeing. So because of this, we're taking in a lot of information about what we're seeing. You have two different pathways, okay, that will carry different information. So the dorsal pathway will go to the parietal lobe. The ventral pathway goes to the temporal. Okay, so again, dorsal is going to be parietal. This is going to be temporal. So I'm just going to remind us of that. So dorsal pathway going to the parietal lobe, that's associated with the interpretation of motion. Okay, remember parietal lobe is where somatosensory cortex is found. So that kind of makes sense. Ventral pathway is associated with the temporal lobe. That's going to be colors, what the shape or form of an object is, how you might recognize one person versus someone else. So if you damage basically part of the temporal lobe or this pathway, you could possibly be able to see something that's round, red, small, looks smooth, but you wouldn't be able to associate it as an apple. A lot of other brain regions, of course, are associated with memory, how you make decisions on what you see. A lot of it is, I don't want to say higher brain centers, those multimodal areas that take into account what you've seen in the past and how that might help you induce or know what to do about a situation that you're currently seeing better. So there's a lot that go into how we interpret what we see. The brain can be very tricky. It can make you think you're seeing things that you're not. Okay, so what we see here is pretty complex. So, you're watching someone surf. So your brain is taking in several different pieces of information. Okay, so the color and the shape of the board, okay, that information is being processed by one part of the brain. The person moving, them falling, that's being accessed by another portion of the brain. As far as all these little steps, what happens, I really didn't get into detail like that. This I definitely didn't. Let's see. We did talk about the sodium channels, photoreceptors, glutamate, that information you need to know. For the photopigment, just memorize this order. So basically, when the stimulus light, okay, is going to hit your cone, first isomerization is going to happen. That basically is producing the pigment. Bleaching is when the pigment's no longer there. Regeneration is it resynthesizing it. More details as far as that gets into, do not worry about that. It's way more detailed than you're going to go. Again, several review questions will help you. So I would definitely review those. And again, in class tomorrow, we will start with the ear.